There are two categories of photoreceptors in the retina of the eye. The rods, which generate impulses in dim light conditions, and the cones, which require brighter light to initiate impulses. Since the human retina has an estimated 125 million rods and only about 6 million cones, the rods outnumber the cones about 20 to 1. Although most parts of the retina have a mixture of rods and cones, the central part of the posterior portion, in an area called the fovea centralis, is composed of cones only. Impulses generated by cones result in color vision and sharp images. Looking directly at an object projects the incoming light rays onto the foveal centralis of the retina. Peripheral vision, resulting from impulses generated in the regions of the retina which lie adjacent to the fovea centralis, is not as crisp as central vision for the simple reason that peripheral parts of the retina contain fewer cones, and the rods are non-functional at moderate to bright light levels. Even in dimmer light conditions where rods can generate impulses, the interpretation of those impulses by the brain results in black and white images which are not sharp and clear.
Hi, I'm Mike Shively, and this is Introduction to Anatomy and Physiology. We're talking about the eye. We began it in a previous session. Do any of you have any questions about things we talked about last time? Amber?
Could you go over again like the layers of the eyelid?
Layers of the eyelid. We went through that kind of quickly right at the end. If we'll go to the overhead camera for just a second and imagine this being an eyeball, kind of inside view here, with the eyelid coming down, upper eyelid coming down like this, and the lower eyelid up like this. Put some eyelashes on here for reference. I'm not sure what this is, but it could be an eyeball if we want it to be. Let's come in from the outside. There's some skin located on the very outside. That's the outer layer. And then within the central part of an eyelid here, there's actually some skeletal muscle tissue forming the orbicularis oculi muscle. I'll write that down here. Orbicularis oculi. And when that particular muscle contracts, because it's a sphincter muscle that goes all the way around the upper eyelid and down around the lower one, it tends to close the eyelids or pull the eyelids closer together. Finally, the lining tissue on the inside of the eyelid, right next to the eyeball, is what we talked about last time as conjunctiva. It's a modified mucous membrane that lines the inner sides of the eyelids, and then it comes down onto the surface of the eyeball. So from the outside in, Heather, it's skin on the outside. There's a little connective tissue proper. Then the next major structure is orbicularis oculi muscle, and finally conjunctiva. Let's write that word again, because it's kind of a strange word to a lot of us. So 1, 2, and 3. Those are the 3 layers from out to in. Any other questions from your reading or anything that we talked about last time? Susan?
Could we go over the components of the lacrimal apparatus again?
Sure, certainly.
Thank you.
Let's just list them here. The lacrimal apparatus, and we talked about these last time, but we kind of flew through them a bit. We've got the lacrimal gland, and that secretes lacrimal secretion, or tears. It's an exocrine gland located on the upper and lateral aspect of the eyeball. It's way back, way underneath the eyelid, too far to try to find, so don't go looking for it. And then when it secretes the tears, or lacrimal secretion, those tears flow freely over the surface of the eyeball and are finally collected into some little tiny openings that are on the medial aspect of the edge of each eyelid. Now, each opening is a lacrimal punctum. And there's one on the upper eyelid and one on the lower eyelid. Let's go to the regular camera for just a second. I know you've had this experience. If you get in front of a mirror, you don't even have to be alone in the bathroom for this one. Pull down your lower eyelid a little bit, and right here in the corner, right next to your nose, you remember seeing that little dot, that little dark dot that's right on the surface there of the lower eyelid. That's the inferior lacrimal punctum. Now, there's one on the upper lid in about the same location. You have to be a little more athletic in front of the mirror to see that one, but that one is also visible. But it's easy to see the inferior lacrimal punctum. So going back to the overhead camera for just a second, there's actually two of these. There's an inferior lacrimal punctum and a superior lacrimal punctum. Those are just the openings. The little teeny tiny channels that carry the lacrimal secretion away from those openings, each channel is a lacrimal canaliculus. And there's two of those, of course. A superior one and an inferior one. Both of those empty into a central collecting area called the lacrimal sac. And it's very, very tiny. And finally, the fifth and last part of this lacrimal apparatus is the nasolacrimal duct. Now, each eye has all of this. So there's a left version of this and a right version. So those are the components of the lacrimal apparatus. Lacrimal gland, two puncti, an inferior one and superior one, two lacrimal canaliculi, a superior one and an inferior one, lacrimal sac, I didn't finish my sac, did I? And then nasolacrimal duct. Those are the components. Yeah, Bryce?
Now, before you mention the eyelashes, are those just to make people look pretty, or is there...
Well, they have some function. Is to keep larger things out of the eye. I don't know if you've had the experience of, you know, when you got really big stuff coming at your face, the eyelashes do have a little protective nature in protecting the eyeball from some of the particulate matter, the bumblebees and the bigger things that might get in there. Any other questions? Eva.
Is it possible to force air backward through that sequence of things for the lacrimal apparatus you just mentioned? I've got a little brother who likes to plug his nose and blow really hard and blow bubbles out his eyes.
So he holds
really
his
nasty.
nose and pressurizes the whole system, and he can get little bubbles of air to actually come out of these lacrimal puncti. Yep. Yeah, and you know, if you might want to see some of these late night talk show hosts. That sounds like one of those things that they would like, but that is entirely possible. Matter of fact, I think I saw one time where somebody did something with some milk in regard to this. It actually got white-colored milk to come out here by pressurizing the whole system backwards. Yeah. Remember, the purpose now of the nasolacrimal duct is to carry that excess lacrimal secretion down into the nasal cavity, where normally, at normal rates of production of lacrimal secretion, the excess is dried by evaporation. Now, the lacrimal secretion is normally dried by evaporative processes within the nasal cavity, just by normal respiratory movements. But if we get in an emotional state where we're disturbed and crying for that reason, or perhaps have irritation of the eye because we have a grain of sand, then the lacrimal gland can go into hypersecretion and produce lacrimal secretion at a rate that's too fast for evaporative processes to dry it out. So that's one of the reasons why our nose runs when we cry, for example. One other thing that I wanted to talk about for just a second, remember when we were drawing the parts of the eye, making our little drawing in a previous session, we said that the lens, and I'm going to put this plastic model of a lens back up here for the overhead camera to take a look at. When it is developing, it has a blood supply that comes in from the back side of the eyeball. So there's blood vessels that plug into the back of it. And then those normally degenerate, and because there's no blood supply to a mature lens, it's avascular. However, the broken up pieces of those degenerating vessels will sometimes float around in the vitreous. And I don't know if you've ever had the experience of looking at a light-colored background, white or maybe the sky, and you'll see these little, they're called floaters, these little things that kind of float down, you know, and you can kind of see them, but then they kind of flicker around, you know, and it's a little bit bothersome. You don't notice them on a dark background, but you do on a light-colored background. And those are actually remnants of those ciliary vessels that came into the back of the lens and nourished it during the developmental process. Well, today we want to talk about how the eye functions. And we have several different subjects to cover in that regard. I want to talk about aqueous humor. Remember, that's the watery solution that fills the anterior chamber and the posterior chamber of the eye. And of course, those two chambers are connected through what opening? What's that opening that joins them? The pupil. So we've got an anterior chamber and a posterior chamber that contain this aqueous humor. And if we go to the overhead camera for just a second and let this be the cornea, sclera coming on around, we won't draw the rest of the eyeball, but if this is the iris right here, go ahead and put the ciliary body in the lens in this eyeball just so we have a reference. The aqueous humor is manufactured by some cells on the back side of the iris. I don't even know the names of those particular cells. They're cells that secrete aqueous humor as a transudate from blood, and that aqueous humor flows very slowly through the pupillary opening into the anterior chamber, and it's finally reabsorbed from through some little tiny channels that are located about the junction of the cornea and sclera. And those little channels used to be called canals of Schlemm, and you'll see that in a lot of physiology texts. Their proper name for those is scleral venous sinuses. So produced by some cells on the back side of the iris, flows through the pupillary opening into the anterior chamber, and reabsorbed through some little tiny openings. Again, canals of Schlemm or scleral venous sinuses is the more proper name for those. And of course, that excess aqueous humor then goes back into the circulating bloodstream. Now, sometimes the aqueous humor's reabsorption is interfered with, and these little reabsorption channels do not work properly. The aqueous humor is still being produced, so it's still being produced, it's not being reabsorbed properly, so what happens then is the intraocular pressure in the eyeball rises, and that can lead to a degenerative condition of the eyeball. Does anybody know the name of the condition? Eva?
Is that glaucoma?
Glaucoma. And one of the scary things about glaucoma is that you can actually have it going on in your eye and not be aware of it. So that's one of the reasons why an annual eye exam is highly recommended, because one of the things that they do in an annual eye exam is to check for intraocular pressure. Adam?
Does that increased intraocular pressure just cause deformation of the eyeball, or can you actually have a rupture?
No, it's not going to go to the rupture state. What will happen is the increased pressure will cause some changes in the lens and then on back in the retina and start causing some degenerative changes. But those kind of things begin to happen before you feel any pain or anything. So you will be unaware of the pressure when it starts rising. All right. So let's look at a graphic that'll summarize what we just said. Aqueous humor is secreted slowly but continuously. Flows through the pupillary opening into the anterior chamber from its site of production in the posterior chamber, and then is normally reabsorbed through the scleral venous sinuses, the quote, canals of Schlemm. Now, if you do have glaucoma, if you do have that condition, there's two ways that it can be treated. One of the ways of treating it is to give some drugs that slow the rate of aqueous humor production down. And that will work in some of the milder cases. If it's more severe, they may have to do some surgical procedures in order to create some artificial drainage channels. Now we want to talk about changing the pupillary diameter. How do we make the pupil bigger and how do we make it smaller? Now, first of all, why do we want to do it? Why do I want to make my pupil bigger? Adam?
To adjust for light conditions.
If light, and what conditions under what conditions would the pupil normally dilate?
If it's darker or less light, the
If
pupil
we're in
will
low light
dilate.
conditions, if we're in real low light conditions, the total amount of light striking the retina will be inadequate to cause the photoreceptors to generate impulses, so we'll dilate the pupillary opening to allow more light into the back part of the eyeball. And the way that we do that, let's go to the overhead camera. I'm going to draw a circle here. I'm going to say that's the pupillary opening. I'm going to draw another circle out here and say That's the edge of the iris. And of course, the eyeball would be clear out here, something like this. So this kind of looks like a target, you know, but today this inner circle is not going to be the bullseye, it's going to be the pupillary opening. Within the iris, radiating like wagon wheel spokes from the pupillary margin out to the base of the iris are smooth muscle fascicles. They go all the way around, and these constitute the pupillary dilator muscle. They are smooth muscle. They're under the control of the sympathetic division of the autonomic nervous system. And I hope that you can understand that if these muscle fascicles contract, they will pull the pupillary margin out toward the base of the iris, which is going to make the pupillary opening larger in diameter. Let me write that down. Pupillary dilator muscles. I'm going to write PUP, period, dilator muscle. I'm going to write SYMP to remind myself that it's under the influence of the sympathetic division of the autonomic nervous system. Also located within the structure of the iris is some smooth muscle tissue that's in the form of a hula hoop ring or a sphincter. It's located just outside the pupillary margin, goes all the way around in a circular conformation, and that is the pupillary sphincter muscle, pupillary sphincter muscle. I'm going to write PUP, period, and then sphincter.
Did you say that was smooth muscle or skeletal?
Both of these are smooth muscle, Susan. Good question. And this one's under the influence of the parasympathetic division of the autonomic nervous system. So I'll just write PARA in parentheses to remind myself that that's under that influence. So these are smooth muscle structures, and one of them, when this one contracts, of course, the pupillary opening dilates. When this one contracts, the pupillary opening constricts. Any questions about those? Okay, just in case you couldn't read my writing, let's look at a graphic where we have that written down. To change the pupillary diameter, we can contract the pupillary dilator muscle, make it bigger in diameter. And so this has sometimes been called the radial muscle of the iris because the smooth muscle fascicles radiate out from the pupillary opening like wagon wheel spokes. The other muscle to make the pupillary opening smaller is called the pupillary sphincter muscle. It's also been called the pupillary constrictor muscle, put that in quotes, because that's in fact what it does. Bryce?
Now, when you squint, does it affect those muscles at all?
When you squint, like in order to see something, you're working real hard to see it. What you're actually doing is changing the shape of the eyeball, which helps change the focal length, if you will, of the lens, to help get light rays focused properly on the retina. And we're going to talk about that in a second.
Okay.
So hang on to that one. All right. Ciliary muscle changes the thickness of the lens. Now, we mentioned previously that the lens needs to be thin if we're looking at something 20 feet or more away. If we're closer than 20 feet, the lens needs to be thicker, and the closer the object, the thicker the lens needs to be. So we need to be able to change the diameter. We're talking about front to back diameter of the lens, okay? We need to be able to change it. Now, let's go back to the overhead camera and look at this little plastic model of a lens one more time that I've brought here. We're looking at the front of it now. As viewed on edge, it's thicker in the center and thinner on the edges. But actually attaching it, coming off the edges of this structure, are these little proteinaceous fibers called zonular fibers that actually attach the edge of that lens to the edge of the ciliary body. Now, the ciliary body, remember, was a structure that was part of the vascular tunic. And buried out in that ciliary body, there is a ring of smooth muscle tissue that goes all the way around. It's called the ciliary muscle. So let me label that, ciliary muscle. And to understand how this lens changes thickness, we need to understand that the lens is made out of an elastic protein material such that it tries to ball itself up into a more or less spherical configuration. The only reason that it is pulled out to where it's thinner on the edges and thicker in the middle is because there's enough natural tension on all these zonular fibers to pull that lens out into a relatively thin configuration. Well, go back to the overhead camera now. When this ciliary muscle contracts, this ring of smooth muscle tissue out here constricts down to form a smaller circle. And what would that do to the amount of tension on these zonular fibers if everything came toward the center? Eva?
It'll relax them a little bit.
It'll relax those zonular fibers, and the lens has a natural tendency to ball up or thicken. And so what will happen under those conditions is it'll get thicker from front to back. From front to back, the lens will get thicker. When the ciliary muscle contracts, the lens gets thicker because the amount of tension on these supporting zonular fibers is reduced. Now, when we relax the ciliary muscle, the natural elastic recoil on all these zonular fibers will pull this lens, pull on the edge of the lens, top, bottom, sides, and everything in between, such that it'll pull the lens out sideways again, and the lens will get thinner from front to back. So it's kind of a backwards thing that you have to remember here. You contract the ciliary muscle, and the lens gets thicker. You relax the ciliary muscle, and the lens gets thinner. Let's look at a graphic that will summarize what we just said here. When the ciliary muscle contracts, ciliary body is constricted into a smaller ring of tissue, smaller circle. The tension of the zonular fibers is decreased, and the natural elasticity of the lens causes it to ball up or thicken, get thicker from front to back in the center. And the next graphic summarizes something else that I want us to remember. A thin lens is needed for viewing distant objects. 20 feet or more away, we need the lens to be basically as thin as it will get. And anything that is closer, we need the lens to be thicker. So when you read, have you noticed that when you read for a long period of time, your eyes get, quote, tired? Have you noticed that phenomenon? And the reason for that is simply you have to contract your ciliary muscle continuously to look at printed words. So you've got to keep the ciliary muscle contracted, keep that lens thick in order to read. So sometimes it's useful when you're reading to what? Look up and look at something distant for a little bit to give that ciliary muscle a bit of a break. Somebody had a question a second ago. Adam?
Is that also why if you bring things very close to your eye, you get kind of an ache or a pain in your eye? Is that because that muscle's contracting?
If you can keep them in focus, you know, you're really having to contract the ciliary muscle very vigorously in order to allow that lens to get as thick as it'll get. Now, in a previous session, we did a little exercise where we brought things in. There's finally a point where you can't keep it in focus anymore. And what's happening is basically you've got the lens as thick as it'll get, and it needs to be even thicker in order to focus that object, and you just can't make it any thicker. Eva?
Yeah, I'm wondering what makes it so people need like reading glasses or have nearsightedness. They can't see
let's
things
talk
up close.
about nearsightedness and farsightedness right now. Let's go back to the overhead, and let's think about the things that a light ray has to go through in order to get that light back to the retina, which is on the back of the eyeball here. There's the optic nerve. And I'm just going to stick a lens right here. We're not going to bother with all the niceties of trying to draw the iris in there. Well, I guess I can draw a little bit of an iris, a little bit of a ciliary body here. Put some zonular fibers in here. All right, now when a light ray comes in, goes all the way through the eyeball like that. First thing that light ray has to go through is a little tiny lacrimal secretion, or tears, on the front of this cornea. And then it goes through the cornea, the thickness of the cornea, that's transparent, right? And when it's in this anterior chamber, passing through the pupillary opening, and in the posterior chamber, what medium, what substance is that light ray in? Aqueous humor. So first we went through lacrimal secretion, then we went through cornea, then we went through aqueous humor, then it goes through the lens, then it goes through vitreous humor. Finally, it goes back here and strikes the retina. Now, we haven't talked about the retina yet, but there are actually 10 layers to the retina. And guess how many layers that light has to go through before it reaches the rods and cones? Has to go through 8 of those 10. The rods and cones form the 9th layer moving outward. So the light ray has to go through 8 layers of the retina before it actually even reaches the rods and cones. So you can see this incoming light ray has to go through a lot of stuff before it excites a rod or a cone. Now, the purpose of changing the thickness of the lens is to get the light rays that are coming in from different objects, to get them bent enough that they come to a focal point right in one particular place on the retina. The place that we normally put that focal point or that image is called the fovea centralis. When you look straight at something, the lens and a little bit the cornea are supposed to bend the light rays enough that they come to a focal point right on that fovea centralis of the retina. Now, if everything is working right, this is what happens, and you have what we call normal vision. However, if the light rays are being bent too much, then we might have that focal point someplace in front of the retina. Maybe it's right up, the retina's clear back here and it's happening clear up here. If this is what's happening, if the focal point, if the light rays are being focused at a point in front of the retina, we have a condition called myopia, or nearsightedness. I'm just going to write myopia equal near for nearsightedness. Focal point is in front of the retina. On the other hand, if the cornea and to a greater degree the lens are not bending the light rays enough, the focal point might be clear back here someplace behind the retina. And we call that condition where the focal point is behind the retina, we call that hyperopia. And that's sometimes called far-sightedness. Now, both of these give us blurry vision. Nearsightedness and farsightedness both give us blurry vision. But simple hyperopia and simple myopia are relatively easy visual deficits to correct if they're not too severe. But they're easy to correct because all we have to do is add corrective lenses in the form of either a biconvex lens shape or a biconcave lens shape. Biconvex, I mean thicker in the center than it is on the edges, or biconcave, I mean thinner in the center than it is on the edges. These would be like the glasses lenses or the quote grind or prescription of contacts. And we can correct myopia, this condition right here. Here the light rays are being bent too much, so we need to spread them out a little bit. We'd need this kind of a lens because when light goes through this kind of a lens, the light rays tend to diverge. So this kind of a lens will correct myopia. And for hyperopia, for farsightedness, the light rays are not being bent enough, and so we need this type of a lens, a regular magnifying glass type that's thicker in the center and thinner on the edges. Because this kind of a lens, when light rays go through it, those light rays that are coming in a parallel fashion like this tend to be converged to a focal point. Okay? So what happens, you add this out in front in the form of glasses, then basically you can bend the light rays a little bit with this, a little bit with the cornea, a little bit with your own lens, and you can bring them hopefully to a focal point right on the retina. And of course, the particular grind or prescription, quote unquote, that's required depends on how far back your focal plane is in the case of farsightedness, or how far up your focal plane is in the case of myopia or nearsightedness. Now, I'll tell you something that's kind of interesting. As we age, eyeballs tend to get longer physically. And part of that, particularly as we get older, from 40 on up, our eyeballs get a few millimeters longer from front to back. In other words, they go from more marble-shaped to more egg-shaped, from front to back. And that's a normal aging phenomenon kind of related to the reabsorption and degeneration of the periorbital fat body, which is a deposition of fat in the back part of the bony orbit. So a lot of young people that start out with normal vision, as they age and their eyeball gets longer, the focal point still stays right here, but the eyeball backs away from it. They tend to get myopia because their focal point tends to be in front of the retina. So a lot of normal people end up with myopia in older age. And so if you've already got myopia as a young person, if you're already myopic, I've got some bad news. It's going to get worse. But if you're hyperopic right now as a young person, if you're already hyperopic, have farsightedness, because the eyeball does tend to get longer, right now your focal point is back here behind the retina, and as the eyeball lengthens, you'll bring your retina back to kind of match where your focal point is. So I got some good news for those of you who have farsightedness. It's going to get better in all likelihood with time. And of course, corrective lenses are employed to deal with the things for the here and now of what's going on in your eye. Now, that's simple hyperopia and simple myopia. There are situations where we have different focal planes in different parts of the visual field. Some of this can occur because the cornea has a flat place on it. You ever get one of those grocery carts at the grocery store? It's got a flat place on one of the wheels, and you go floppity, floppity, floppity down the aisle. Okay.
Every time.
Yeah. You know what happens? That happens from going out in the parking lot and riding them and then putting on the brakes and skidding sideways. That's the way to do it. At any rate, if we have a visual field that has different focal points and different focal areas in different parts of it, in other words, this part's in focus but this part is not, or this part's in focus but this part up here is not, when you look at something, that condition is called astigmatism. How many of us are astigmatic to a certain degree? Am I the only one in here? No, we got, there's 3 of us. Okay. Those are very difficult visual deficits to correct because it's usually a flat place on the cornea or a flat place on the lens that causes that. And so you have to have a very, very carefully prescripted lenses in order to get those particular parts of the visual field corrected without interfering with adjacent parts of the visual field. Anyway, that's what astigmatism is all about. Different focal links in different parts of the given visual field. Sure.
I was just curious, what does laser surgery involve when they're correcting that?
When they do laser surgery, Tyler, what they're doing is they're changing, look over here on the overhead for just a second. Let's say this is the cornea, and here goes the sclera away from that. They're making some little cuts in the surface of the cornea without going all the way through it. And depending on how they orient these cuts, they can end up to make the cornea bulge out more than it did before, or to even be flatter than it was before after these heal up. Mm-hmm. And by changing the radius of curvature of the cornea, now the cornea is not a major player in focal length, but it does play a little bit of a role. Because it is curved, it does play a role. So by changing this, then they can change a little bit what your focal length is back here and either correct simple versions of myopia or hyperopia. And it beats wearing glasses all the time and seems to be pretty well accepted and relatively safe procedure, as evidenced by the number of people that have had it done and had their vision significantly improved. So you're changing the curvature on the cornea by making some little cuts with laser light type scalpel that don't go all the way through. All right, let's review some of the things that we've just said. Let's go to a graphic here that summarizes hyperopia, farsightedness. Either the eyeball is too short or the lens is too thin, and the light rays are not getting bent enough. Anyway, the bottom line is we're getting a focal point that's behind the retina, and we want that focal point to be right on the retina. And we're going to correct this with a converging or magnifying lens, one that is thicker in the center and thinner on the edges. On the other hand, myopia, next graphic, also called nearsightedness, the eyeball is either too long or the lens is too thick And the problem is the focal point is in front of the retina. And we're going to have to correct that with a diverging lens, one that is thinner in the center and thicker on the edges. All right, I want to talk a little bit about that retina now. I believe we made the point in a previous session that it's a mixture of rods and cones that serve as the photoreceptors. Most of the retina is that mixture. There's a lot more rods than there are cones. And a couple of things we need to know about rods versus cones. First of all, both of them are photoreceptors. When you shine light on them, what do? Generate an impulse. That impulse is then carried through some sensory neurons in the optic nerve back to the occipital lobes of the cerebral hemispheres, where the impulses are interpreted as vision. So our eyes don't really see per se. All they do is generate impulses. The actual perception of those impulses occurs back in the occipital lobes of the cerebral hemispheres. Now, at normal light levels, like we have in this studio right now, only the cones are working, because the rods have to have very low light levels. In normal light levels like we've got here, the rods are being overdriven and they are non-functional. Only cones are working in normal light levels. And I think we made the point previously that cones give us all colors, all color vision, including the colors of black and white, incidentally, are due to impulses from cones. And cones give us sharp, crisp images when those impulses are interpreted. Rods, on the other hand, give us only black and white, no colors. The images are not very sharp. You know, when it's dark and you can barely see shapes, that's when your rods are working full force and you don't have any cones. If you can't see color, you haven't got cones working at that particular light level. And so when I think we made the point before, there's a certain level of light that's kind of halfway between bright light and low light, where some of the rods and some of the cones can both be working at the same time. So it's not an all or none phenomenon. But if we've got color vision, it is due to cones. There is a central area right in the back of the retina called the fovea centralis, where it's just 100% cones. And that's where we tend to focus an image when we're looking straight at something. We bring that image right in onto that particular area, and it's all cones. It's no rods in the mix in that particular part of the retina. So if you look at me up here, you know, I'm in color. I think we're in color. And if you look at me, maybe if I scoot over here to this side, you can see this skeleton over here on this side. If you're looking at me, the skeleton is a little bit fuzzy, but it's there, right? Or if you look at the skeleton, I'm a little bit fuzzy, but I'm here. Now, the reason that peripheral vision is a little bit fuzzy at normal light levels is because when you look straight at something, the image comes into the fovea centralis, got lots of cones there. They're all generating impulses, color vision, nice, clear, crisp images. When you're looking at something in your peripheral vision at reasonable light levels like we have here, the image of that object is falling onto a peripheral part of the retina. And in that peripheral part of the retina, well, we have a mixture of rods and cones, and the rods are doing nothing. So you've got fewer pixels on the screen that are operating for your peripheral vision at normal light levels. Let's take that another step. Let's go out on a starry night and let's look at the dimmest star that we can see in the sky. You've had this experience, I'm sure. If you haven't, go out tonight if it's a starlit night. I don't know what the moon's doing. You know, I haven't checked that lately. But go out and look at the sky and find the dimmest star that can be seen. Now, have you noticed that when you look straight at that dimmest star, what's it do? It fades out. You can't see it. Why? Because when you look straight at it, you're projecting its image where? Onto the fovea centralis, and what's there? All cones. And extremely dim stars don't put out enough light to be picked up by cones. Now, some of the brighter stars put out enough light that some of the cones will be stimulated. Anyway, let's go back to this dim star. You look straight at it, it disappears. If you look just a little bit to the side, that dim star will appear in your peripheral vision. So you say, there it is, and then you look back and it's gone. And you look away, and there it is, and then you look back and it's gone. It's kind of a maddening experience. I want you to have it. How many of you have had that experience already? Okay, 4 or 5 of us have had that. Tyler, go out tonight. Maybe you can have some fun yourself. Get a life. All right. All right. Let's take a look at a graphic that summarizes the layers of the retina. We mentioned there are 10 of them total. The outermost layer of the retina, the one that's closest to the back of the eyeball, okay? The outermost, so the light ray would strike this particular layer last, layer number 10, is pigmented. And then layer 9, coming forward, the 9th layer that the rods and cones, that the light would actually strike, is the layer of rods and cones, okay? And then there are 8 other layers in front of the rods and cones. So the light has to go through these 8, then it strikes this, and then it goes on and strikes this pigmented layer. Now, physiologists don't know for sure if the light stimulates the rods and cones on the way through them the first time, or whether it goes out and strikes this pigmented layer and is reflected back into the rods and cones. But there's some pretty good physical evidence that probably the latter is what happens, because the rods and cones are turned around and pointed not at the incoming light rays, but they're pointed at the back of the retina. So we think what happens, the light goes all the way through the rods and cones, strikes the pigmented layer, and is reflected back into them. Now, this pigmented layer is very well developed in animals that are active at night, nocturnal animals. And you've been driving along at night and seen a skunk or a deer or something like that, and you can see their eyes shine in the headlights of your car because the light is going in, striking that pigmented layer, and then is being reflected back out toward you where you pick it up. People do not have quite as well a developed pigmented layer, and as a consequence, normally all you get when you shine a flashlight in somebody's eyes is some angry comment about don't shine that light directly into my eyes. All right, let's go ahead, Adam.
Is it the pigmented layer that causes red eye in photos?
In the prints, in the old color prints, particularly on some of the older film, flash, you'll see red-looking eyes, because what'll happen is it'll go back to the back of the retina. I think actually what's happening is reflecting the back of our retina has kind of a reddish yellowish color when you look at it through an ophthalmoscope. And I think that's just being reflected back out at us. And the camera then picks that up and records it on the film. Let's take a look at a graphic that summarizes the things we need to remember about rods. They generate impulses in dim light only. The impulses are interpreted only as black and white images, no color, and the images are not particularly sharp or crisp. If we take a look at that same information on cones, look at the next graphic. This summarizes the cones. They require more light than the rods. Their impulses are interpreted as color vision, all colors, including black and white, and the images are sharp and crisp. I want to look at another graphic. This next graphic shows you the actual reaction that's occurring. One of the chemicals in the rhodopsin, or the photochemical, is a material called cis-retinine. Now, both rods and cones have this cis-retinine. And under the influence of light, the cis-retinine is converted into trans-retinine. That causes the impulse to be generated. That change of cis-retinine over to the trans, which again is triggered by light, causes the impulse to be generated, and then that impulse is synaptically transferred to sensory cells, sensory neurons that carry the impulse onto the cerebral hemispheres. Now, that trans-retinine can be reused if it's regenerated with ATP back to cis-retinine. So we can recycle this retinine back and forth. Light converts it from cis to trans. It requires ATP to convert it from trans back to cis. Now, at normal light levels, normal, reasonable, moderate to bright light levels, cones can regenerate this back from trans to cis. And so cones continue to operate at normal light levels. At moderate to bright levels, though, rods cannot do this part. They simply can't do this part of the reaction unless the conditions are pretty dark. So that's the reason that rods don't work in bright light. They get what we call bleached out. They cannot regenerate their cis-retinine, so all the photopigment is stuck over here in the trans form, and only until we get those rods into a dark environment can the rods regenerate back to here. And those of you who've had the experience of having the room lights turned off in a fairly bright room, turn off the lights, and if there's a dim light down the hall, you can still, in a few moments, you can pick up images and be able to see some things. When the lights first go off, you don't see anything for a little bit. And then slowly the rods begin to regenerate the cis-retinine, and then you can start seeing at least some shapes with your rods. Any questions about that phenomenon? Okay, there are actually 3 different kinds of cone cells. Now, when we say different kinds, we mean cone cells that respond to red light, cone cells that respond to light in the blue part of the spectrum, and cone cells that respond to the light in the green part of the spectrum. So we have red, blue, and green cones. The reason that we have those different color designations on the cones is simply because the particular protein that the retinine is hooked up with responds to light in those 3 different parts of the visual spectrum. Some individuals have a color blindness problem where they don't have all 3 of these cone types, or maybe they have them but there's something faulty about the protein material, so they have a color blindness problem. They have a certain part of the color spectrum where they can't pick up the subtle differences in different shades of that particular color. If we look at a graphic that summarizes these 3 kinds of cone cells, we see we have the red, blue, and green. That means they respond to red light, they respond to blue light, they respond to green light. They differ in the proteins with which the retinine is associated, and that's why we have the 3 different designations. And they absorb light in different regions of the photospectrum. Now, those of us who have normal vision have all 3 kinds of cone cells, and all of them are working correctly. But people with, well, let's take a look at a graphic that summarizes what we just said there. This next graphic. Normal color vision. We have all 3 kinds of cone cells, and they all work okay. We're called trichromats in the, by an ophthalmologist or an optometrist. If we have normal color vision, we're a trichromat. We can see all 3 of these colors in the visual part of the spectrum. If we're missing one type of these cone cells, then there's different names for those. If we're missing just one of the 3 types, we're called a dichromat. So if we go to this next graphic, a dichromat that happens to be missing the cone cells that respond to red, the light color red, then they have a condition called protanopia. So they've got the blue ones and the green ones, they don't have the red ones. If they're missing the green ones and just have the red ones and blue ones, that condition is called deuteranopia. And if they're missing the blue ones, and just have the red and green, then they have a condition called tritanopia. So those are just names for particular kinds of color blindness. I used to worry about these folks at traffic lights, and wonder, well, I wonder if that guy's colorblind, and does he know when it's red and when it's green? And then somebody pointed out, well, duh, that's a Utah expression that means what? But of course, everybody knows that. Well, the red light's on top, and then the yellow, and I said, well, yeah, but sometimes they take the traffic signal and they flip it over into horizontal format, so you've got them all side by side. But a lot of people that have some kind of a color vision deficit, they're not totally colorblind maybe. They can see some different colors, but there may be certain shades of certain colors, that they don't operate well or can't differentiate all that well. Eva?
I'm just wondering, you said we have cones that respond to red light, blue light, and green light. Why is it we can see all kinds of different colors? So why is it everything's not just red?
When I said
blue.
red, blue, and green, it means that the optimal color, they optimally respond to a certain wavelength, but if if you're on one side or the other of that wavelength, they'll still respond to some. So we can see, and it's basically with those three colors of light, you can kind of mix and match those to get almost the whole visual spectrum of light. So I don't know what the exact frequency of the wavelength, that is optimal for the red ones or optimal for the blue ones or optimal for the green ones, but those things are known. That's how we can see different colors within that. It's kind of like, there's the taste just as modalities are just salt, sweet, bitter, and sour, but we can taste more kinds of things than just those due to the sense of smell and so forth. Bryce?
I've never seen one, but like in a 3D movie, I guess they give you the glasses with the red and green lenses. And it's supposed to make stuff pop out at you. What makes that happen?
I'm not sure how that works. I went to a couple of those once, and I can remember it was kind of an interesting experience because the first time it was a cowboy and Indian flick, the old kind, and the arrows were flying, and they had arrows that looked like they were coming, and I was ducking down behind the seat because it really gave you the impression that it was happening. Maybe, have any of you ever been to Universal Studios? You've been there, Amber. Did you get in some of those funky rides where you get in this little thing and you feel like some unbelievable things are happening, and you're going to crash and so forth. And in reality, it's all, you're being fooled because of the images that they're projecting in front of you.
Yeah.
It's one of those kind of interesting experiences. Is there another question over here? All right, let's see. We've talked about how to change the focal length by changing the thickness of the lens. I did not give you the word for that, and that's called accommodation. Let's go back to the overhead camera and look at that. This is simply a 25-cent word, which means, hey, we're going to change the thickness of the lens. And remember that to make the lens thicker, we have to do what to the ciliary muscle? Contract it or relax it?
Contract.
Contract it. Now, I want to emphasize this because it seems just the opposite, like it ought to be the opposite. And to make the lens thinner, we have to relax the ciliary muscle. Now, don't forget that. Any questions on this subject? Bryce?
Like if you do a lot of reading, can that make your eyesight worse because you work that muscle so much that it tends to stay a little more contracted?
You know what?
No?
You can't. It doesn't hurt. It's exercising. Now, as parents, we tell our children these little lies. There's a whole series, and these have all been cleared with Heavenly Father, so it's okay. But one of the stories we tell them is that if you read your comic books or whatever under the covers at night with a little dim flashlight, that you'll damage your eyes, and then you'll be blind, when you're an adult. And the fact of the matter is simply not true. Reading in inadequate light levels, while it may be difficult to see the print, doesn't damage your eyes in any way. A lot of reading doesn't hurt your eyes at all. It exercises that ciliary muscle, which you have to contract. Now, incidentally, when you start to get older, the lens loses some of its elasticity. And when you want to make it thick, by contracting the ciliary muscle and reducing the tension on those zonular fibers, it doesn't get as thick as you want it to. And when you want it to get thin so that you can see distant objects clearly, it doesn't get as thin. It kind of gets stuck halfway in between. Instead of going from thick to thin, it goes from not quite as thick to not quite as thin. So it'll still move a little bit, but it doesn't get as thick as you want it to get, and it doesn't get as thin as you want it to get. And this condition is called presbyopia. Presbyopia. Let me put that word on the overhead so everybody can see it. And the definition is simply loss of elasticity of the lens. And so with that lens not being as elastic, it doesn't get as thick as it should, doesn't get as thin as it should, then you tend to have visual deficits in both at both extremes, and you basically just have trouble with distant vision as well as with close-up vision when you have this condition. Adam?
Along with all of this, when watching TV at 4:30 in the morning and infomercials or AM talk radio, they have advertisements for systems where you can improve your eyesight by doing a series of exercises. Is there any basis to that in fact, or?
When we squint by kind of squeezing in with our eyelids, we squeeze on the eyeball and thus change its shape a little bit. And with extremely mild visual deficits, you can move the back of the retina a little bit one direction or another and perhaps change a blurry situation to one that's crisper or sharper. By and large, we're not talking about something that's going to help people that are severely myopic or hyper.
It's not going to work for me.
Probably not. Incidentally, when we say someone has 20/20 vision, does everybody understand that that's just some nomenclature that means an object that's 20 feet away, if I have normal or 20/20 vision, an object that's 20 feet away looks like it's 20 feet away. If I have 20/30 vision, then something is 20 feet away. I can only see it as good as I could see, a normal person can see it if it were further away. So if the second number is bigger than 20, then you have worse than normal vision. Maybe you're 20/40, 20/60, 2200, 2400, whatever. If the second number is smaller than 20, like 2015, then an object that's 20 feet away, you can see it as well as a next person could if it were only 15 feet away. So that's what those numbers mean as far as visual acuity. So, hopefully you've got 20/20 or better, 20/18, 20/15 or something like that. Birds of prey have phenomenal vision because they can take an object, a little meadow mouse, and they're hovering up here. Maybe a red-tailed hawk could be up there 2,000 feet, and he can see that mouse moving in the grass, something we wouldn't be able to pick up unless we were just a few feet away.
Are they looking for movement, or are they looking, or they actually see the mouse?
They actually see the mouse. Well, unless the grass is thick on top of them. I think we need to quit now, so we'll hold up. They're rolling, and they'll cut out our... Oh.